This poster is for the Discover Student Research 2024 Symposium. My name is Noah Menard, and I work in Dr. Shog's lab. My poster is on the development of an online and stop-flow LC benchtop NMR system. One of the reasons why we're approaching this is that complex mixtures are hard to resolve when being tested inside an NMR. Coupling it to an LC, however, would give us the ability to separate out mixtures and hopefully resolve some of these issues. There have been previous iterations of LC-NMR in the past, but they involve large, high-field NMRs. The coupling is difficult, and overall, the system is extremely expensive. On the other hand, benchtop NMRs are becoming more and more prevalent, and as the technology progresses, we're starting to see higher and higher field strengths available in the magnet. This makes the idea of revisiting the LC-NMR idea much more viable. To start with, we needed to connect the two systems. We connected them physically, and then to verify that the connections were in place and the systems were functional, we injected acetonitrile using a mobile phase of undeuteраted methanol. Our initial tests showed that we had successfully made our connection, and that we were seeing the strongest NMR signal, or the peak maxima, at 11.9 minutes. This let us know that as the system was working, when we should start looking for particular times to begin stop flow testing. However, acetonitrile is not detectable by a photodiode array, which we use in our LC, so we needed to move on to other samples. Our initial analytes were ibuprofen, acetaminophen, caffeine, and warfarin. These have a variety of different peak types in the NMR, peak shapes, and peak locations, giving us a good spread to analyze our data over and verify the system. Online testing requires the NMR to be, of course, continually on throughout the test, and we need low flow rates so that the NMR can resolve the data. It also requires a high analyte concentration and consequently has also low sensitivity. To start with, we then began testing at a very low flow rate of 0.01 mils per minute in order to increase the resolution and determine the peak maxima of the NMR time-based scan. Over time, we were able to detect these peak maxima across all 4 samples. They held true for all 4 samples. And from this, we were able to determine the lag time between the PDA and the strongest, highest concentration of analyte within the NMR cell. From there, we were able to move on to stop flow testing. This improves resolution. We can use lower concentration samples, and we can use faster flow rates. From the previous calculations, we know when to stop the instrument after a peak appearance time. And then we can begin analyzing the sample. This was successful. And using this, we can then confirm that our initial ideas were correct, and we could now move on to complex mixtures. Our future plans now are going to be moving on to column separation and individual analysis of each one of the analytes within the complex mixture. We plan to reduce the amount of analyte needed, improve our flow rates to something more reasonable, and determine the more technical conditions of the optimal flow rate of the LC compared to the time needed to equilibrate in the NMR. Future work continues, and so far results look promising. Thank you.